Well, we'll go ahead and get started. So we'll continue our discussion about epigenetics, and today we will focus on a new group of disorders called the Mendelian disorders of the epigenetic machinery, which are genetic disorders with epigenomic consequences. So the objectives for today are to become familiar with Mendelian disorders of the epigenetic machinery, to define broad categories of disease within the group, to recognize key phenotypic features, to recall inheritance patterns and disease mechanisms, to discuss the roles of target genes and altered epigenetic marks in disease pathogenesis, to recognize the unique potential for therapeutic manipulation of this group of disorders, and then to identify them as genetic disorders with epigenetic consequences, including genome-wide DNA methylation patterns that are specific for each disorder. So the Mendelian disorders of the epigenetic machinery, which are also called chromatin-modifying disorders, also called chromatinopathies, are a rapidly emerging group of conditions. Back in 2014, Hans Bjornson and I wrote a review about these disorders. There are about 28 genes involved. In 2019, we wrote the review with this figure here, and there were about 70 genes involved, and more recent counts, depending on how you define these disorders, are upwards of 85 to 100 disorders. And prior to 2014, we didn't even know this was really a group of disorders. Some of them were known, but we didn't know there was a whole group of them. So the 70 genes responsible for these conditions are shown in the wheel and are divided into categories based on whether they encode enzymatic writers of epigenetic or chromatin marks, enzymatic erasers of these marks, non-enzymatic readers of these marks, or enzymatic chromatin remodelers that alter chromatin structure. And in many cases, the enzymatic writers, erasers, and remodelers also have reader domains, and those that do are shown shaded in gray and have been referred to as dual function components. The most common phenotypic features of these disorders are intellectual disability and growth abnormalities. Intellectual disability occurs upon disruption of about 83% of the genes and is indicated by blue shading around the periphery. Intellectual disability is defined as significant limitations that originate before the age of 18 in intellectual functioning and adaptive behavior, affecting many social and practical skills. Growth abnormalities occur in about 71% of these conditions, or are associated with mutations in about 71% of the genes, and are indicated by orange shading around the periphery. Growth abnormalities can be in the form of growth deficiency, which is a bit more common, and overgrowth. And these two are defined as, deficiency is defined as height and or head circumference greater than 2 standard deviations below the mean for age. And overgrowth is defined as height and or head circumference greater than 2 standard deviations above the mean for age. And about 87% of the intellectual disability genes, when disrupted, also cause growth abnormalities. In addition, there are many tissue diverse manifestations in addition to these most common features. And these include other neurobehavioral findings like hypotonia, which is one of the most common also, anxiety, autism, ADHD, seizures, and then non-neuronal features include musculoskeletal features like scoliosis, craniofacial dysmorphisms, eye findings, structural anomalies of various organs, and in some cases, immune dysfunction. So in support of this, Bucas et al. Have shown that these genes are highly co-expressed across tissues and among individuals. So high co-expression across tissues and among individuals could help to explain another characteristic of these genes, which is their high dosage sensitivity. So when all the genes are considered, they have a PLI score, 80% have a PLI score of greater than 0.9. So the PLI score is the probability of being loss of function intolerant, and the range is from 0 to 1, and anything greater than 0.9 is highly intolerant to loss of function. If you look at just the intellectual disability genes, 91% have a PLI score of greater than 0.9. So in line with this high dosage sensitivity, the vast majority of conditions are inherited in an autosomal dominant manner, and that's indicated by the filled, dominant inheritance is indicated by the filled circles around the periphery. There we go. And autosomal is indicated by the A in the inner circle. Certainly there are exceptions to this. Most of these disorders result from de novo mutations with a mechanism of haploinsufficiency. So about 77% of conditions with intellectual disability are autosomal dominant, and about 7% of conditions with intellectual disability are autosomal recessive. So there are two broad groups that we think of with these disorders, and they kind of separate out as we talked about on Friday. We separate them based on whether they affect the histone post-translational modification system or the DNA methylation system. And as we talked about, I'll just point out, as we talked about last week, both of these groups, as you may recall, have their own distinct sets of writers, erasers, and readers. So here's an example of the epigenetic machinery that affects a particular histone mark. The histone mark is shown with these red hexagons, and the teeter-totter indicates a particular gene. The epigenetic machinery consists of enzymes that place the marks, called writers, enzymes that remove the marks, called erasers, and then these non-enzymatic reader proteins. And here, epigenetic marks, as we talked about on Friday, determine whether chromatisms are open or closed. And then this determines whether genes are turned off or on. So if a writer places a silencing mark, this leads to a more closed chromatin state and gene silencing. And vice versa, if an eraser removes that silencing mark, then this can lead to more open chromatin and gene expression. But that's a very simplified view of one mark, one promoter. As we talked about on Friday, this is not really how things work. In the normal state, there are many components of the epigenetic machinery replacing many different marks and removing many different marks and otherwise modifying these marks. And so it's a much more complicated picture. So again, we think of these disorders as genetic disorders with genome-wide epigenetic consequences. So in the normal situation, you have various components of the epigenetic machinery working together to allow some genes like gene 1 to be expressed and other genes like gene 2 to be silenced. So gene 1 has writers of activating marks, erasers of silencing marks, readers of these marks, and chromatin remodelers that alter chromatin structure. And the net effect is a more open chromatin state in gene expression. Genes like gene 2 have writers of silencing marks, erasers of activating marks, and readers of these marks, with the net effect of a closed chromatin structure and gene silencing. In the case of a Mendelian disorder of the epigenetic machinery, if, for example, a writer of an activating mark is not functioning properly, then it doesn't get localized properly to genes like gene 1 to set up this particular chromatin structure and allow for gene expression. And rather what can happen then are you can have this abnormal chromatin structure where you have instead silencing marks and the silencing machinery leading to this aberrant closed chromatin state and gene silencing. But that's the direct effect of the loss of the histone methyltransferase here. But there are also indirect effects. So if the silencing machinery here is mislocalized to gene 1, then other genes in the genome, like gene 2, which is supposed to be silenced, does not, these genes have an aberrant chromatin state that's sort of an indirect effect. Because if all the silencing machinery is sort of tied up elsewhere, there may not be enough for it to localized where it's supposed to, and this can lead to an abnormal chromatin state that's more open and abnormal gene expression. So with these, there are many direct and indirect effects, and these are happening in many genes throughout the genome, causing broad epigenomic changes. And one of these changes is DNA methylation. So here you can see that originally in the normal situation, gene 2 had DNA methylation at the promoter, but in the abnormal situation, this DNA methylation is no longer present at gene 2, but rather is now present at gene 1. And so we now know that this DNA methylation at CpG dinucleotides can be indirectly altered, directly and indirectly altered in the setting of these disorders. And there are many disorders which have these characteristic DNA methylation profiles, which we'll talk about in a little while, showing that this is indeed the case. So the affected target genes then may determine the phenotype in these disorders. So the hypothesis is that genetic mutations in the epigenetic machinery cause disease by disrupting epigenetic marks and target gene expression in trans. Now this is distinct from what we learned about on Friday, because we were talking about imprinted genes where the epigenetic changes occur and they just disrupt gene expression in cis. But here we're talking about disrupting gene expression in trans. So to illustrate this, in the setting of a properly functioning writer of a silencing mark, which normally silences genes A, B, and C, but genes D through H are expressed. In the setting of a Mendelian disorder of the epigenetic machinery, when this writer is not functioning properly, what we can see is these A, B, and C are no longer silenced, and instead genes D, E, and F are abnormally silenced, and genes G and H don't change. So if gene A promotes growth, gene B is involved in learning or forming memories, and gene C is involved in craniofacial development, then these are the phenotypes that will be affected directly. Also, if genes D, E, and F are involved in certain neurobehavioral features, heart development, or muscle tone, these things will also be affected. And so this is kind of how you can get a multi-system disorder in the setting of one of these disorders.
So I guess I understand how it might lose the ability to find a gene, but what is sort of the mechanism that it hits an off-target gene? Is it like a DNA binding domain in the writer that gets altered? Like, how is it interfacing with this gene but not that gene?
Right. So it depends. So it depends on where the mutation is in the component of the epigenetic machinery. But also, remember we talked a bit on Friday about the shifting of chromatin, right? So like I used the example of the oncohistones, where if you have a mutation in one of the oncohistones, then you get one of the components of the epigenetic machinery can then bind there and be kind of stuck there essentially, and then it can't put the marks where they're supposed to be, and so then other marks that are inhibited by that mark can then kind of encroach in. So it's kind of, there's not one answer to that question. It kind of depends on what the effect is from the actual pathogenic variant or mutation. But the idea is sort of like from the basis of what we talked about on Friday, like that these marks interact with one another and these components interact with one another. And so if you have a mutation in one that causes a problem with its localization, then there can be these broader epigenetic changes. Does that make sense? All right, great. So I wanted to illustrate the concept that target genes play a key role in pathogenesis, as I just kind of explained theoretically, and that these are trans effects as opposed to cis effects that we saw with imprinting disorders. And to do this, I'm going to use two different examples, one of Soto syndrome and one of Kabuki syndrome 1. So this is our wheel again, so we're going to talk about Soto syndrome, which results from pathogenic variants in the NSD1 writer. And that writer places a particular epigenetic mark called H3 lysine 36. And here is a schematic of the NSD1 gene. Soto syndrome is caused by heterozygous de novo mutations in this gene. It's inherited in an autosomal dominant manner. Their de novo and unique mutations predominate. You can see that this is an older slide, but truncating mutations occur throughout the gene, and missense mutations tend to occur in the functional domains. The mechanism is one of haploinsufficiency. While Soto syndrome is due to alterations in NSD1, there are a variety of ways which it can occur. About 75% are associated with intragenic mutations, like you see here. About 5% can have intragenic deletions, and about 10% have whole gene deletions, so 5q35 deletions, and then the other 10% is unknown. Most likely the other 10% is that they don't actually have Soto syndrome, but they have some related condition whose gene has not been identified yet, or they were diagnosed with Soto syndrome many years ago because this is one of the disorders that's been known for some time, and then they were never retested, and so maybe the gene is known, but they don't have a diagnosis. So pathogenic variants in NSD1 cause Soto syndrome. So individuals with Soto syndrome have a pretty striking phenotype. And the most striking characteristic is their generalized somatic overgrowth, which is manifest by tall stature and or macrocephaly. They also have mild to moderate intellectual disability, which is kind of an unfortunate combination because they're very much larger than their age, but they have intellectual disability, so they're expected to act older, but because they look older, but they have intellectual disability. They also have characteristic neurobehavioral profile that includes severe anxiety, impulsivity, attention deficit hyperactivity disorder, or ADHD, and obsessive compulsive disorder. And they constantly are asking the same question over and over again. Socially, they can be quite awkward. They have characteristic facial features with this long face, a tall, broad forehead with a prominent and we call prognathic chin. They often have these widely spaced eyes that can be down slanting. And they can have this malar hypoplasia or sort of thinning of the cheeks with this redness that we see, that we don't really understand why they have that. And their facial features are most prominent at about 4 years of age. They can also have advanced bone age, hypotonia, joint laxity, scoliosis, seizures, various eye findings, cardiac anomalies, and renal anomalies. They also have characteristic brain MRI findings. They're not specific findings, but they're very characteristic for Soto syndrome, and so when you see these together in an individual who has features of Soto syndrome, they can almost be diagnostic. So they have thinning of the corpus callosum, which is the part of the brain that connects the right and the left side. They have enlarged lateral ventricles and prominent subarachnoid spaces. So the subarachnoid spaces are this sort of dark area between the bright white, which is the skull, and then the brain parenchyma. So they have an increased space there. And that's thought to be because their skull is growing faster than their brain. But sometimes it unfortunately gets called as brain atrophy, but that's not what it is. So these are very characteristic brain MRI findings. So there was a group who found two siblings with a phenotype similar to Soto syndrome, including macrocephaly, with or without tall stature, intellectual disability, characteristic behaviors like ADHD, brain malformations, and some features suggestive of Soto syndrome. And these individuals were found to have biallelic variants in APC2. APC2 is expressed in postmitotic neurons and is involved in axon guidance and neuronal migration. So they hypothesized that these individuals were so similar to Soto syndrome because maybe APC2 is a downstream target of NSD1. They made an APC2 knockout mouse, which was quite similar to the patient's. The mice were found to have relative macrocephaly, abnormal head shape, impaired neuronal migration, and impaired learning, memory, and behavior. Also importantly, they had a lot of the brain findings that I showed on the brain MRIs from individuals with Soto syndrome. They had thinning of the corpus callosum, which you can see here, which is magnified here, compared to control mice, and that's quantified here. They also had the enlargement of the cerebral ventricles, which you can see here, compared to the control animals, and again, that's quantified here. This group performed knockdown with RNAi and showed abnormal neuronal migration when they targeted APC2 or NSD1, shown here. So you can see in the control, it's a very organized pattern, but with APC2 or NSD1 knockdown, you see this disorganization, abnormal neuronal migration. And then importantly, NSD1 mediated neuronal migration defect was actually corrected with overexpression of APC2. And so they concluded that APC2 is responsible for, this APC2 is downstream of NSD1, and suggested that it was responsible for some features, including intellectual disability, abnormal behaviors, macrocephaly, brain malformation, but may not be responsible for other features of Soto syndrome. Yes?
Has anyone done chip-seq experiments for that H3K36 mark and the APC gene?
Not that I'm aware of. I'm not aware of much happening after this paper on this, but that would be the next step. And people have done chip-seq for H3K36 dye and trimethylation in other systems, but not this one that I'm aware of. But that would be the next step, so good call. Okay. So that's sort of one way of trying to identify target genes, is identifying another gene that has mutations in which have a similar phenotype, and then you look and see what's upstream, what's downstream. And there are other ways of identifying target genes as well, and I'm going to illustrate that by looking at Kabuki syndrome 1. So Kabuki syndrome 1, this is the mutations in KMT2D cause Kabuki syndrome 1 and cause growth deficiency by disrupting histone marks and target gene expression. So how did we figure this out? So Kabuki syndrome is more common form of Kabuki syndrome and is an autosomal dominant disorder resulting from mutations in the KMT2D writer that places H3 lysine 4 trimethylation and monomethylation. So it's the deficiency of an activating mark. So individuals with Kabuki syndrome 1 exhibit the main features of intellectual disability, growth deficiency or short stature, and distinct craniofacial dysmorphisms. So they have these interesting eyebrows where they have flaring of the eyebrows and notching. They have these everted eyes, so the lower lids are everted, which means that they're sort of flipped outward a little bit. They have these long palpable fissures and very long eyelashes. They have this broad nasal tip and then a short columella, which is the part of the nose that connects the nasal tip to the face. And so that short columella gives this appearance of a flattened facial profile. And here you can see evidence of their growth deficiency or short stature. So height is on the y-axis, and on the x-axis is age. And you can see that individuals with Kabuki syndrome, so this is the 50th percentile, this dark line in the middle. And this is the lower limit of normal for height for age. You can see that individuals with Kabuki syndrome tend to have short stature. So Hans Bjornson and Hal Dietz created this mouse model of Kabuki syndrome 1, in which they replaced the catalytic domain with a beta-GO cassette, creating a heterozygous loss of functional allele in the KMT2D gene. And Hans's group went on to characterize the neurobehavioral and immune phenotypes, which we'll talk about a little we'll talk about the neurobehavioral phenotype later. Our group looked at the growth deficiency phenotype. So we noticed that when we did x-rays of the mice, the Kabuki syndrome mice were smaller than the wild type mice, and that included total body weight and total body length. We also observed when we performed high resolution micro CT of the skulls that Kabuki syndrome mice had this flattening of the facial profile, and they had this sort of almost shortening of the AP diameter, flattening of the facial profile, and kind of brachycephaly, which is flattening of the back of the head. And so this was reminiscent of Kabuki syndrome. They also had shortened long bones. So both male and female mice had shortening of those are femurs there. So both had shortening of the femurs. Interestingly, so many of these features are consistent with Kabuki syndrome. And interestingly, with respect to the craniofacial phenotype, there's an area of the skull that's responsible for the growth of the AP diameter of the skull, meaning the growth in this direction. And that is the area that would be suspected to be affected when you see these findings on micro CT. So just to kind of give you a little overview of the process that we think is disrupted in these mice. So endochondral ossification is the process by which long bones grow, and it's a process by which that one particular area in the skull also grows. And so here's a schematic of a long bone right here, and then this is the growth plate. And what we're showing here is the chondrocytes in various stages of development. So there's the resting zone, the proliferative zone, the hypertrophic zone, and then the calcifying zone. And the way that long bones elongate is first the cells are in the resting state, they receive a signal to proliferate, and they form these long columns. And then as they proliferate, then they begin to differentiate in hypertrophy. So that's kind of these white cells where they begin to hypertrophy. And then the osteoblasts kind of come in and replace this cartilage with bone, and that's how the long bones grow. And then we think that, the way we think about this is sort of the bone that's formed here is trabecular bone. So we next evaluated the growth plates in the Kabuki mice, and we found that the heights of the proximal tibia growth plates were measured. When we measured them, the heights were found to be increased in the Kabuki mice compared to controls in both sexes, and this is quantified here. We also found that this involved both the proliferative and the hypertrophic regions. And it was due to increased cell numbers, and in females it was also due to increased cell size. So what we're really trying to get at here is what is the cellular phenotype in these mice, so that we can then hone in on that cell type and better understand the molecular events like changes in gene expression. So we thought it was interesting that the growth plates were expanded when the bones were actually shortened. That was a little counterintuitive. So we looked at the skull region that also grows by this process and found that, in fact, the growth plates were also expanded, and the quantification is shown here. And so this suggested that this was a real finding. So we wanted to get at the mechanism and identify a cell type of interest, but it's difficult to study that in the living animals, so we turned to a more manipulatable system, the ATDC5 chondrocyte, or it's an ATDC5 cells. They're a mouse mesenchymal cell line that, when induced to differentiate, go through various stages of chondrocyte development, so proliferation, hypertrophy. And so we generated Kabuki syndrome 1 mutations in the cell lines using CRISPR gene editing. So the top is a biallelic deletion. The middle is a deletion on one allele and a deletion of an amino acid on the other. Just a single amino acid in the set domain, which is the catalytic domain. Sorry, I can't see that very well. So sorry, this is the wild type, my bad. this is the single amino acid on one allele and deletion on the other allele, and then the bottom one is the biallelic deletion. When we looked at this, when we stained with this Alcian blue staining, which is a stain that recognizes proteoglycans in the cartilage or in the chondrocytes, we found that the cells from the mutants seem to be differentiated more precociously, and this is quantified here. So the more severe the mutation, the sooner these chondrocytes were differentiating, and we sort of saw like a dose-dependent increase in differentiation with worsening mutation. So we performed genome-wide transcriptome profiling in Kabuki syndrome 1 and control chondrocytes, and this is a volcano plot, so it shows the log 2 of the fold change, so the change in gene expression on the x-axis, and negative log 10 of the p-value, which is significant on the y-axis. So anything in red is significant. And so what you can see is that we found that SOX9, which is a master regulator of chondrogenesis, and COL2A1, which is a collagen found in that's made by chondrocytes, were both upregulated in the mutants compared to the controls, and SHOX2, which is a gene that is homolog of the SHOX gene in humans, was downregulated. And we found the same results in undifferentiated cells compared to chondrocytes. So we were able to then kind of narrow this down to see if we could find any interesting target genes of interest. So when we looked at genes that were both altered in chondrocytes and in differentiated cells, and then if we narrowed that down to just those that had a greater than 4-fold change in gene expression, we found 74 named genes. We were more interested in those we wanted to identify a direct target of KMT2D, so we were more interested in those that were downregulated because KMT2D places an activating mark, so you would expect direct targets to be downregulated if you lose that mark. And so we were quite interested in the SHOX2 gene. We confirmed that it was in fact downregulated by qRT-PCR. And when we looked back at the mouse growth plates, we found that in fact the SHOX2 expression at the protein level was reduced in the Kabuki syndrome mice. So we then wanted to see if there was a change in the epigenetic marks there. So we looked at H3 lysine 4 trimethylation at the promoter region. And what we found was that in the mutants, this mark was reduced compared to control mice. And when we looked then at gene expression of SOX9, that master regulator of chondrogenesis, and we wanted to see if overexpression of SHOX2 could impact this pathway. So we found that, in fact, when we overexpress SHOX2, so this is the control with no SHOX2 overexpression. This is the mutants with no SHOX2 overexpression. But then when we overexpress SHOX2 in the mutants, we found that the gene expression level of here was reduced, but not all the way to control levels. We also looked at COL2A1, which is a marker of chondrogenesis that the cells express, and we did not see a change there. And when we looked at the cellular phenotype more broadly using the Alcian blue staining, we found maybe a little bit of a rescue when we overexpressed SHOX2. So what our conclusion was from these studies is that SHOX2 may be a new target of KMT2D, maybe a direct target gene, but there are many others. And by just reactivating this target gene, that was not sufficient to rescue the cellular phenotype, suggesting that a single target gene may not be sufficient to rescue a cellular phenotype in these conditions. Moving on now, so here from what I just showed you, target genes were identified for both Sotos syndrome and Kabuki syndrome 1, but single target genes didn't seem to be sufficient to explain the full phenotype or rescue that phenotype. So the therapeutic implication of that is maybe we need to target the epigenetic machinery rather than individual target genes.
Is it possible that there's a temporal switch, that it's not just on, that it's on for a certain amount of time before it's recognized by a eraser or something like that?
Yeah, that's certainly possible, for sure. The balance hypothesis is, I'm going to kind of use the next set of experiments to explain the balance hypothesis and show that it's plausible and testable, and that this offers unique opportunities for therapeutic manipulation. So the balance hypothesis, which Hans Bjornson and I put forth a few years back, is that both writers and erasers of the same mark are present at particular target genes, and that the correct balance is important for properly turning on and off genes. And this balance determines the presence or absence of a particular mark and whether the chromatin is more closed or open. And so according to this balance hypothesis, the Mendelian disorder of the epigenetic machinery can disrupt the balance of chromatin at target genes when a writer or an eraser of a particular mark is not functioning properly. And then this can lead to changes in levels of the mark, changes in whether the chromatin is more open or closed, and thus whether target genes are turned on or off appropriately. So next I want to look at Kabuki syndrome 1 and Kabuki syndrome 2 together, side by side. So one question is, do common epigenetic consequences at target genes underlie similar phenotypes in Kabuki syndrome 1 and Kabuki syndrome 2? And so Kabuki syndrome 1 and Kabuki syndrome 2 are Mendelian disorders of the epigenetic machinery with highly similar phenotypes. So here's an individual with Kabuki syndrome 1, individual with Kabuki syndrome 2. They both have intellectual disability and developmental delay, a characteristic neurobehavioral phenotype. They have growth deficiency and or short stature. They have very common craniofacial dysmorphisms that we talked about before. They have low muscle tone, immune dysfunction, structural anomalies of organs, and hearing loss and eye findings. So Kabuki syndrome 1 results from deficiency of a writer of an activating mark, as we talked about. So the deficiency is of KMT2D, which places an activating mark, the H3 lysine 4 trimethylation mark. Kabuki syndrome 2, however, is actually an X-linked condition and results from mutations in KDM6A, which is an eraser of a distinct silencing mark. And so how could both of these lead to essentially the same phenotype, though we know there are some differences? So our hypothesis was that the commonality may lie in the fact that both kind of tip the balance of chromatin toward a more closed state. So deficiency of the writer of an activating mark, deficiency of the eraser of a silencing mark. So, immunofluorescence experiments in Kabuki syndrome 2 mice have shown an increase in H3 lysine 27 trimethylation in the granule cell layer of the dentate gyrus, which is what would be expected with deficiency of an eraser of a mark. You would expect that mark to be increased, and then, again, the balance hypothesis, you would expect that to lead to a more closed chromatin state. And in Kabuki syndrome 1, Hans Bjornson's group showed that the KS1 mice have decreased H3 lysine 4 trimethylation in the granule cell layer of the dentate gyrus. So again, deficiency of the writer of this mark, you would expect there to be a reduction in that mark. And that's shown in the pink staining. So in their paper, and this is the paper that I sent along for discussion next Friday. So they reasoned that if the balance hypothesis was true, in Kabuki syndrome 1, with deficiency of an activating mark, which would tip the balance toward a more closed state, inhibition of any mark that would sort of tip the balance back in the opposite direction could restore the balance of chromatin. So, what they did is they treated mice with Kabuki syndrome 1 with a histone deacetylase or HDAC inhibitor to attempt to restore the balance. So they showed that the H3 lysine 4 mark, shown in the bright pink here, was reduced in the granule cell layer of the dentate gyrus of the hippocampus in these mice compared to controls. And this was associated with a reduction in neurogenesis. So this is the green staining shows double cortin, and it is reduced in these mice. And so that suggests a neurogenesis defect. So when they then went on to perform some behavioral testing in the mice, platform zone crossings is what they measured. And this is something that you measure when you perform the Morris-Water maze. So the Morris-Water maze is a test of mouse visual spatial learning and memory, where the mice have to use visual cues to navigate to this platform to be able to get out of the water, which they do not like. And so they found that the Kabuki syndrome 1 mice have visual spatial learning defects similar to patients with Kabuki syndrome 1. However, when the mice were treated with increasing doses of AR42 and HDAC inhibitor, they showed that there was restoration of the neurogenesis in the dentate gyrus and restoration of the H3 lysine 4 trimethylation mark. And they were also able to show an improvement in learning and memory on the Morris-Water maze for the mice. And this was done in young adult mice, suggesting that this is something that could be used as a potential treatment. And they've gone on now, because this is a bit of a non-specific way to do it, because HDAC inhibitors are kind of broad and they hit a lot of genes, but they've gone on to use both a dietary manipulation called the ketogenic diet that generates a substance in the body, beta-hydroxybutyrate, which can act as an HDAC inhibitor, and found similar results. And then they've also used kind of the opposing component of the epigenetic machinery by inhibiting the opposing eraser, and have shown that that also can show similar effects. So this, is very, very encouraging. And now Jackie Harris, who's our colleague, is working on moving forward with clinical trials to try to see if this would actually work in humans. All right, so now moving on to Weaver syndrome. So we're going to talk now about Weaver syndrome, which is highly related to Soto syndrome, which we talked about before. And this is a deficiency of the EZH2 writer of the H3 lysine 27 trimethylation silencing mark. So Weaver syndrome and Kabuki syndrome 2 are opposing disorders. So we just talked about the fact that Kabuki syndrome 2 results from mutation and leads to deficiency of an eraser of the H3 lysine 27 trimethylation silencing mark. And that these individuals have growth deficiency. In Weaver syndrome, this results from a deficient writer of this same mark and leads to overgrowth. So opposite molecular and phenotypic findings. So individuals with Weaver syndrome exhibit striking somatic overgrowth. So this is the average height of individuals in the UK over time, or by age. You can see that individuals with Weaver syndrome are well above that average height. And when we look at their actual height standard deviations, they can be anywhere from +2 to +7 standard deviations above the mean for height. So this is a 12-and-a-half-year-old girl with Weaver syndrome who's about 6 feet tall. And I diagnosed this individual with Weaver syndrome, and she is also about 6 feet tall and wears a size 15 shoe at age 17. So in addition to the striking overgrowth, these individuals have intellectual disability, distinct and distinct characteristic facial features. So they have this kind of round face, these full cheeks, and this kind of stuck-on appearing chin. They can also have a tall, broad forehead and widely spaced eyes. So we generated a mouse model of Weaver syndrome. And so Weaver syndrome is distinct from some of the other conditions in that the vast majority of individuals have missense changes as opposed to loss of function changes in the gene. And so we generated the most common Weaver syndrome missense mutation, which is the arginine 684 cysteine mutation in mice. And we found that in this is present in the catalytic set domain. So this is the domain responsible for placing the H3 lysine 27 trimethylation mark. And while heterozygous pups survived, homozygous pups did not, but we were able to isolate mouse embryonic fibroblasts and show that this mark is actually reduced globally in homozygous mutant nuts. And there's really kind of a dose-dependent decrease in the mark as you, with the worsening mutations. However, EZH2 itself was stable. We next looked at the phenotype of these mice, and really what we want to do, and this is a common theme in this lecture, in this field, is we want to understand the phenotype and see if the mouse model is a good representation of the phenotype, but also be able to kind of hone in on a cell type of interest and a cellular phenotype, because we know that epigenetic marks can differ between different cell types, and so we want to hone in on a cell type that's actually relevant to the phenotype, and then try to understand what's going on at the molecular level so we can understand pathogenesis, but also design therapies. So here we saw that our female Weaver mice had overgrowth or increased body weight. But when we looked more closely at the skeletal phenotype, we see that the mutant mice have enlarged bones. So you can see here the Weaver syndrome mutant mice have significantly larger cross-sectional areas of their femurs, which is the thigh bone, and we saw that in both male and female mice, suggesting a skeletal overgrowth phenotype. So in order to get at the actual the cells that were responsible for the phenotype, and better understand the molecular events, we used a technique called dynamic histomorphometry. And in this technique, 2 compounds that incorporate into newly formed bone were sequentially injected into the live mice, calcium, which is shown in green, and alizarin red, which is red. And this was done at the time when bone growth was rapid, but before we could actually detect overgrowth. And then 2 days later, the mice were euthanized. And what you can do with this technique is you can actually see how the green and the red labels get incorporated. And then and then you can measure the distance between the green and red labels and divide that by time and get a rate. And so what we were getting at was the mineral apposition rate, which is a measure of osteoblast activity. And so when we did this, these are the actual images from our mice. And this is looking at the periosteum, which is the outer surface of the bone, and then the endosteum, which is this inner surface of the bone. And we found that the endosteal mineral apposition rate, or osteoblast activity, was increased in mutants compared to controls, and that the periosteal was increased only in females. That showed that in vivo we had an increase in osteoblast activity in the Weaver syndrome mice compared to controls. So we then wanted to look at cells that we could study in a dish to look at the cellular phenotype. So we isolated bone marrow mesenchymal stem cells from Weaver and control mice and differentiated them into osteoblasts in culture for 21 days. So these cells are basically coming right from the bone marrow, which is the precursors that go on to form the cells that we studied in vivo. So they're a relevant cell type, and they're the precursors to the cells that we studied in vivo. So we isolate those from the mouse bone marrow and then differentiate those into osteoblasts and measured their ability to lay down bone with alizarin red. And we observed increased uptake in alizarin red in the Weaver samples compared to the controls, and that's quantified here. So this suggests that they're behaving similarly to what we saw in vivo. We then were able to take those cells and do RNA-seq and compare the Weaver osteoblasts to controls. And we found that when we looked at volcano plot, they had a distinct gene expression profile where there were 100, so again, log2 fold change or expression levels on the x-axis, negative log 10 at the p-value, or significance is on the y-axis. And everything in yellow is significant. This line indicates the 10% FBR cutoff. So we had 194 differentially expressed genes with 100 downregulated and 94 upregulated. So remember, we're just looking at differences in gene expression, not only direct targets, and so this is why we have both up and downregulated genes. We have direct and indirect targets. So this is the volcano plot shown again, but this time looking at just osteogenesis genes in blue. So all three of these genes, RUNX2, SMAD5, and SP7, are important in osteogenesis. And we suspected that osteogenesis genes may be collectively dysregulated because we saw a lot of them in our gene expression, differentially expressed gene list. So we used the mouse genome informatics database to identify a list of 201 genes that had previously been described as being involved in osteogenic differentiation. And we performed a Wilcoxon rank sum test, and the p-values of these genes were significantly shifted from 179. So 179 of these genes were expressed in our data set. 179 of the 201 from the mouse genome informatics database. So we looked at these genes and compared them to 179 random genes and found that these genes their p-values were significantly shifted from the other genes, suggesting that osteogenesis genes are in fact collectively dysregulated in these cells. And the same for the BMP pathway or the bone morphogenetic pathway. These genes are also collectively dysregulated. So we next wanted to see if we could, now that we had a cellular phenotype that was measurable, and now that we knew that also gene expression in these cells was dysregulated, we wanted to see if we could reverse or prevent the skeletal, the increased laying down of bone or the increased osteoblast activity in cells from the mice. And so remember that EZH2 is the writer of this H3 lysine 27 trimethylation mark. KDM6A is the eraser of that mark. So we hypothesized that if we inhibited the opposing eraser, we might be able to shift the chromatin back towards steady state. So we first looked at the cellular phenotype, and you can see that the cells treated with vehicle are shown here, and the cells treated with this drug are shown here. And what you can see is that in the mutant cells, if you compare the mutant cells, vehicle-treated cells with the drug-treated cells, you can see that there is a reduction in the laying down of bone. And in fact, this laying down of bone is quite similar to the control vehicle-treated samples, suggesting that this treatment could reverse the cellular phenotype in the cells from these mice. So, of course, the next step is to look at gene expression. So we know there was disrupted gene expression, particularly the osteoblast differentiation pathway and the BMP pathway. And so we again performed genome-wide transcriptome profiling with RNA-seq. And we, this is again this is a similar volcano plot to what I showed previously. But this plot shows vehicle-treated mutants compared to vehicle-treated wild type. So it's similar to the untreated mutant versus wild type, which we looked at previously. The osteoblast genes are shown in blue and BMP genes are shown in magenta. And you can note these genes that are highlighted here that are upregulated in the mutant compared to the wild type. And all of these genes promote osteogenesis. This volcano plot compares the mutant cells treated with GSKJ4 versus the mutant cells untreated, or vehicle treated, sorry. This gives the readout of the differentially expressed genes upon treatment with GSKJ4. So note the same 4 genes that promote osteogenesis revert from being upregulated in the mutant compared to the wild type to being downregulated upon treatment with the drug. So they reverse their fold change. And when we look at these two groups in the same plot, so on the x-axis, we have the mutant versus the wild type, which represents the effect of the mutation. And on the y-axis, we have the mutant drug-treated versus the mutant vehicle-treated, which represents the effect of the treatment. And what we found is that 1,045 out of 1,075, or 97% of the shared genes that we noted, reversed fold change directionality. So what that means is that if you look here, genes that are upregulated in the mutant compared to the wild type are downregulated upon treatment, and genes that are downregulated in the mutant compared to the wild type are upregulated upon treatment. So this suggests that we are able to reverse a significant amount of the gene expression. This is quite different than when we looked at the prior study where we were just looking at one target gene. So it does suggest that targeting the epigenetic machinery is actually a better approach than target gene-specific therapy. And so this is just a slide kind of summarizing that potential future therapies. What these could include certainly CRISPR gene editing or targeting certain components of the epigenetic machinery where they need to go. But also we could consider that we can also have a unique opportunity in this set of conditions, because remember from Friday, I showed you those lists of all the drugs that have been designed and tried in cancer that are epigenetic therapies, many of which target these same components of the epigenetic machinery. So we have, this whole sort of other list of drugs that we could try in these genes that would be unique to trying to treat these disorders compared to other things like CRISPR gene editing or gene therapy or things like that.
In the genes that are upregulated prior to treatment, do they recapitulate the phenotypes of the gain of function of those types of genes? So like RUNX2 and fiber dysplasia ossificans, I think. Does that show up as like a phenotype in these patients or mouse models with just an upregulation of expression?
So it's laying down too much bone, right?
Yeah.
So that's what that disorder and phenotype, or the RUNX2 is implicated, or is the genetic cause of fiber dysplasia ossificans, which is, so everybody else knows, you're laying down too much bone in abnormal places, right? So here we have just generalized overgrowth, so they're just laying down too much bone, but not necessarily in the wrong places, but they're just laying down too much bone. So it does kind of match, yeah. All right, so for the last half hour, I want to now talk about how these disorders how we know that these disorders have epigenetic consequences, and mainly we'll focus on genome-wide DNA methylation patterns. And so in order to do this, I'm going to tell you the story of a syndrome that we recently described to sort of illustrate this concept. So we knew before, and we talked about briefly on Friday, that we have the writers, erasers, and readers of the DNA methylation machinery. And previously we knew disorders that impacted the writers of DNA methylation and the readers of DNA methylation. but there wasn't a disorder known to impact the TET genes, the erasers of DNA methylation. So we identified the first neurodevelopmental disorder of the DNA demethylation eraser system, so resulting from mutations in TET3. So just as a quick reminder, we saw this slide on Friday. So the TET enzymes, remember, are the erasers of DNA methylation. They are dioxygenases that oxidize 5-methylcytosine through a series of intermediates, including 5-hydroxymethylcytosine, 5-formylcytosine, and 5-carboxycytosine. The latter two intermediates are then removed and replaced by unmethylated cytosine. So just remember this first step in the pathway. So 5-methylcytosine being converted to 5-hydroxymethylcytosine. That's going to come up in a few minutes. So I just want to tell you the story of how we elucidated this disorder, and sort of how you go from identifying variants in one patient to knowing that this is actually a disorder. So we performed trio exome sequencing on the first family, family 1, and identified biallelic variants in TET3. We then used GeneMatcher. I'm not sure if you've heard about GeneMatcher, but GeneMatcher is kind of a matchmaking service for geneticists and researchers where you can put in variants that you've identified in patients and match with other people around the world who have also identified variants in these genes. And it was designed here at Hopkins by Nara Sobrera, Ada Hamish, and Dave Valley. So we used GeneMatcher to connect with other researchers who had found variants in TET3, and we identified 11 individuals from 8 families with variants in TET3. And this gene was known at that time to be important in embryological development and neuronal functioning. So affected individuals from families 1 through 3 had biallelic variants inherited in trans from carrier parents. However, this seemed to be consistent with autosomal recessive inheritance. However, in all instances, one or both parents of each family had milder manifestations. Affected individuals from families 4 through 8 had monoallelic variants in TET3, and particularly importantly, in family 7, the proband inherited the variant from a similarly affected father. So suggesting we weren't just missing the other variant in the other families, but that it actually could be transmitted as an autosomal dominant condition. And so these affected individuals had highly similar phenotypes that were consistent with other Mendelian disorders of the epigenetic machinery. So they had developmental delay and intellectual disability in all cases, growth abnormalities, hypotonia, autism spectrum disorder or autistic features, movement disorders like tics or other findings, and then seizures and or EEG abnormalities. They also had some characteristic craniofacial features, but you can see they're not as characteristic, for example, as like Kabuki syndrome or Soto syndrome or Weaver syndrome. But they did have, many of them have tall and or broad foreheads, brachycephaly, which is flattening of the back of the head, a long face, a hypotonic face, which is illustrated by the mouth being open, and protruding ears in some cases. So TET3 variants identified in these individuals were novel or rare, and here the monoallelic variants are shown in purple and the biallelic variants are shown in orange. Missense variants in the first paper could be monoallelic or biallelic and mostly occurred within the catalytic domain. And frameshift variants were only occurred in the monoallelic form in the first paper, but could be located anywhere throughout the coding region. And then underlying variants occurred in the last exon. And then this italicized orange variant is now questioned. We're now questioning whether that is pathogenic. So in collaboration with Roberto Bonazzio's group at University of Pennsylvania, we performed functional studies to test the catalytic activity of a subset of the variants using an in vitro TET activity assay. And this is important, when you find variants in a particular gene, because you want to get some idea of what is going on, and if they actually are changing, for example, in this case, a catalytic activity. So TET3 variants or controls were overexpressed in HEC293 cells, and the ability to convert 5-methylcytosine to 5-hydroxymethylcytosine, that first step in DNA demethylation, was measured using an antibody that specifically recognizes 5-hydroxymethylcytosine on dot blot assays. 5-hydroxymethylcytosine levels were measured in cells expressing each of the 5 TET3 variants shown with the asterisks, and compared to 5-hydroxymethylcytosine levels in cells expressing wild type or a known catalytic dead control. Quantification revealed that all variants tested showed reduced catalytic activity similar to the catalytic dead control, with the exception of one, the arginine 752 cysteine variant, which is the variant I said on the last slide we questioned its pathogenicity, and it was outside the catalytic domain and not conserved among TET enzymes. So these data show that most TET3 variants exhibit hypomorphic activity, at least the ones we tested here, in vitro, and suggest that the disease mechanism is one of reduced TET enzyme activity. So reports in the literature have identified these highly sensitive and specific genome-wide DNA methylation signatures in blood associated with a growing number of these conditions and with other disorders, as we talked about on Friday. So we wanted to determine if deficiency of TET3 is associated with a DNA methylation signature. And so I just want to illustrate what these signatures look like. So in a healthy individual, they have a reference DNA methylation signature. So gene A is methylated, gene D is unmethylated, and genes B and C are partially methylated. In this individual, all the components of the epigenetic machinery are functioning properly. In syndrome A, where maybe there's a mutation in a particular writer of an epigenetic mark, it doesn't have to be a DNA methyltransferase, it can be any component of the epigenetic machinery. You might see that gene A becomes fully unmethylated, gene D becomes fully methylated, and genes B and C remain the same. And this is episignature A, which corresponds to syndrome A. In syndrome B, when a distinct component of the epigenetic machinery, maybe an eraser of a different silencing mark, is not functioning properly, you have gene A becoming fully unmethylated, gene B becoming fully methylated, and genes C and D not changing. And this is episignature B corresponding to syndrome B, and so on and so forth. DNA methylation profiling that I'm going to talk about today uses the Invidium DNA methylation array technology. And the way this works is that first, DNA is treated with sodium bisulfite, and this is called bisulfite conversion. And so cytosines that are unmethylated get deaminated to uracil. Cytosines that are methylated remain cytosines. And then through RT-PCR, these uracils become thymine, and the cytosines remain cytosines. So this DNA is then run on a DNA methylation array, which has these site-specific CpG probes shown here. The bisulfite converted DNA is shown in green down here. And so if the former C is now a T, then a red labeled A gets incorporated. If the bisulfite treated DNA, if this base, the former C remains a C, then a green labeled G gets incorporated. And so the amount of methylation at each CpG site is determined by calculating the ratio of the signal intensities for the methylated versus the total methylated plus unmethylated probes. And this is referred to the DNA methylation level or beta value at each site. But it's this pattern of beta values at many sites, hundreds to thousands of individual CpG sites across the genome, that constitutes these DNA methylation profiles or episignatures. So, of course, we were interested in determining whether or not this new disorder had a genome-wide DNA methylation profile, and suspected it would, especially since it directly impacts DNA methylation. So we use these DNA methylation arrays to better characterize the disorder and to determine variant pathogenicity. And there is an actual clinical test that we use in the clinic where we run these DNA methylation arrays and can diagnose patients with disorders or interpret variants of uncertain significance. So here we hypothesized that deficiency of the TET3 eraser of DNA methylation would lead to an overall increase in DNA methylation and have a characteristic DNA methylation profile. So we performed the genome-wide DNA methylation profiling of whole blood using a discovery cohort that consisted of 3 individuals with biallelic TET3 variants, 3 individuals with monoallelic TET3 variants, and 5 age and sex matched controls for each affected individual. We used Illumina's Athg B chip array, which has over 850,000 CpG probes, and subsequent analyses were performed in collaborator Beckham Sidocovic's lab. So hierarchical clustering analysis is shown, and each row represents an individual CpG site, and each column represents an individual patient sample. And these are color-coded based on whether the individual has monoallelic or biallelic variants in TET3 or are an unaffected control. The DNA methylation level or beta value is shown here and ranges from dark blue, which indicates no methylation, to dark red that indicates fully methylated. So the hierarchical clustering reveals an overall increase in DNA methylation and clearly separates the TET3 samples from the control samples, and even shows that the biallelic samples, shown in blue, may have more of an increase in DNA methylation compared to the monoallelic samples. So just looking at the data in a different way, this is a multidimensional scaling plot, which is like principal components analysis. You can see that the biallelic TET3 variants are on the cluster on the far left, and the controls cluster on the far right, and the monoallelic variants cluster in between. And so together these data establish a DNA methylation profile for TET3 deficiency or Beck-Farner syndrome, which separates the TET3 deficient samples and can even further differentiate those with biallelic or monoallelic variants. So we next validated the TET3 epi signature using a distinct cohort consisting of individuals with known monoallelic pathogenic TET3 variants. So these were either missense variants that reduced catalytic activity in our in vitro assay, or de novo nonsense variants. Unaffected family member controls were also included, and the benign missense variant that didn't reduce catalytic activity in our in vitro assay. So here the original discovery cohort samples are shown in faded colors, and the new validation samples are shown in bright colors. And the multidimensional scaling reveals that all samples validated with the new monoallelic pathogenic samples clustering with the old monoallelic pathogenic samples, the family member controls clustering with the age and sex matched unrelated controls, and then this variant that didn't reduce catalytic activity in our in vitro assay is somewhere in between. So another way of looking at the data is by the methylation variant pathogenicity score, which is the probability that a particular TET3 variant is pathogenic. And biallelic samples have scores near 1, monoallelic samples with monoallelic variants have scores between 0.5 and 0.8, and then controls have scores near 0, again with this benign variant that didn't reduce catalytic activity somewhere in between. So we then refined the TET3 epi signature using both the discovery and validation cohort samples and their matched controls, which are shown in faded colors, and used the new signature to test a new cohort, the testing cohort, consisting of biallelic, shown in orange, and monoallelic, shown in purple, variants of uncertain significance. So both biallelic samples, shown in orange, and the 4 monoallelic samples labeled 1920, 23, and 24, clustered with controls, shown in green, suggesting non-pathogenicity. The other 4 samples, 21, 22, 25, and 26, clustered with known monoallelic pathogenic samples, indicating pathogenicity. This sample, shown in black, is unique. So all the other samples had DNA sequence variants in TET3, and we're performing these analyses to determine whether the sequence variants are more likely pathogenic or benign. This sample, however, was not known to have a DNA sequence variant in TET3 and was identified based on the TET3 epi signature when this signature was used to screen a database of over 1,000 undiagnosed individuals. So it pulled out this individual, And this variant clusters with other monoallelic pathogenic variants, suggesting that this individual could have a pathogenic variant in TET3. So subsequent review of the individual's prior exome sequencing identified a monoallelic nonsense variant in TET3 that was inherited from potentially mosaic mother, confirming the diagnosis and illustrating the robustness and diagnostic utility of this TET3 episignature. The methylation variant pathogenicity scores supported the other analyses, and importantly, when we compared this method to other methods that are more traditionally used to classify variants, including the presence or absence of NOMAD, the CAD score, the combined annotation dependent efficiency score, I always forget the last D, and then inheritance and segregation in families, and then whether or not the variant was within or outside the catalytic domain. Using those metrics, we also would classify them similarly. And just to give one example of how inheritance and segregation allowed this, so the orange biallelic samples are children with biallelic variants in TET3, but they're variants of uncertain significance. And then 2 of these are parents of one child and 2 of these are parents of the other child. So in one family, we were able to do segregation analysis, and we found that the individual who has biallelic variants in TET3 also had a healthy, completely healthy sibling who had the same 2 variants in TET3. And so that basically confirmed that that individual's variants were not pathogenic. And so it completely validated this assay for that family as well. So we next wanted to determine whether the TET3 deficient episignature would be able to distinguish Beck-Farner syndrome or TET3 deficiency from other conditions with known episignatures. So we first took a one-against-all approach and compared the TET3 samples compared the TET3 samples to samples from 46 other neurodevelopmental disorders with 38 distinct episignatures and their controls. And we used 75% of samples for training, shown in blue, and 25% for testing, shown in gray. And we found that the samples from the controls, which are shown here, and all other syndromes were distinct, or had scores near 0, indicating that the TET3 episignature was able to differentiate individuals with this condition from individuals with other conditions. But there was one exception, and that's this one right here. So this is Athg, or autosomal dominant cerebellar ataxia, deafness, and narcolepsy syndrome. It's a mouthful. These individuals have hypermorphic variants in DNMT1, a writer of DNA methylation. So their episignatures partially overlap with that of TET3. So we directly compared DNA methylation ranges in samples with TET3 and DNMT1 variants and their respective controls. And so as I said, as I've shown, hypomorphic variants in the TET3 eraser of DNA methylation in Beck-Farner syndrome, and hypermorphic mutations in DNMT1 writer of DNA methylation in Athg would both be expected to cause an overall increase in DNA methylation, and this is what we observed. So compared to controls shown in green, samples with pathogenic TET3 variants, which are shown here in red, purple, and blue, and Athg samples, which are shown in black, have an overall increase in DNA methylation. And then when we looked at the multidimensional scaling plot, we could see that these samples were all clustered separately. So the controls over here in green, the TET3 samples up here in the top left, and then the Athg samples down here at the bottom, and then again, still the TET3 biallelic and monoallelic samples clustered separately. But this shows that they do cluster separately from one another, and that these episignatures can distinguish individuals with these 2 disorders. And then this is just an example to show that this is not only true for our disorder, but it's true for many disorders. So this is a dimension reduction plot of DNA methylation for 15 different syndromes. Each point is an individual, and then each symbol represents a different condition, and those are shown here. So you can see that individuals with distinct conditions cluster separately. So, in summary, Epigenetic mechanisms play a key role in proper gene expression, and disruption leads to disease states. This new group of disorders, Mendelian disorders of the epigenetic machinery, are an important emerging group of conditions that result from germline mutations in epigenetic machinery genes. The most common phenotypic features are intellectual disability and growth abnormalities, and the genes are highly co-expressed. Inheritance is mostly autosomal dominant, and the genes are highly dosage sensitive. Individual direct target genes are important to pathogenesis, but certainly not the whole story. And the balance hypothesis is testable and plausible and suggests that therapeutic manipulation is likely to be successful. And so I hope that I've shown you that these disorders are genetic disorders with genome-wide epigenetic consequences. And then DNA methylation profiles are one example of this and are important in the diagnosis of patients. So this is just my acknowledgement slide, and then this is just a slide from our clinic. If anyone's ever interested in observing in our clinic, we'd be happy to have you. We see patients with these disorders a couple times a month. So thank you, and I'm happy to take any questions. Just references. All right. Great. Is anybody, I don't know if anyone's watching online, any questions?
Thank you so much for the online.
Oh, okay. Thank you.